Human chromosome number 2 is often said to be good evidence for a shared evolutionary ancestry between humans and chimpanzees. However, when one looks at the details more closely, they find it doesn't support the evolutionary model, but instead supports intelligent design. Hello, my name's Jack and welcome to Bowtie Proclamation. In this video, we'll be looking at how evolutionists use human chromosome number 2 to make the case for evolution, and how the details of human chromosome number 2 show that it doesn't actually support the model being proposed by evolutionists, but instead supports intelligent design. With that out of the way, let's get started by talking about chromosomes. Your DNA is broken up into multiple segments called chromosomes. The largest of these is called human chromosome 1, and then the second largest is chromosome number 2. And the numbering system goes on as the chromosomes get smaller and smaller. Excluded from this numbering system is the sex chromosomes, the X and Y chromosomes, as they have different sizes to each other. As this video is about human chromosome number 2, it's about the second largest chromosome in humans. Now, the number of chromosomes will vary from animal to animal. In humans, the DNA is broken up into 23 pairs of chromosomes. In chimpanzees, gorillas, orangutans, and potatoes, the DNA is broken up into 24 pairs of chromosomes. That's one more pair of chromosomes than humans. To account for the discrepancy in the number of chromosomes between humans and chimpanzees, evolutionists have proposed that at some point since humans split off from chimpanzees about 6 million years ago, 2 chromosomes fused together into what we now know as human chromosome number 2. Now, chromosomes have some important structures that we'll need to talk about as well. At the end of chromosomes are structures called telomeres. These protect the rest of the chromosome from a process known as DNA shortening, in which as DNA gets copied, it gets shorter and shorter and shorter. To protect the genes and therefore the health of the individual, these structures exist at the end as a buffer, if you will, of this repeated sequence so that the genes aren't affected. In humans, this repeated sequence is TTAGGG. Another important structure of chromosomes is called centromeres. These are located near the center of the chromosomes and are important for cellular division. Just before a cell divides, the chromosomes are duplicated and meet in the center of the cell. Spindle fibers attach from the ends of the cell to the centromeres of these chromosomes. Then, when the tension in the spindle fibers is just right, the chromosomes move to opposite ends of the cell, so that the new daughter cells from the cellular division will have the same amount of DNA as each other. This structure also has a repeated sequence, although at a much larger one with 171 base pairs. Outside of the centromere and the telomeres is a region that contains the genes responsible for making proteins that the cell will use. And outside of the regions where the genes are is what regulates those genes. To account for the discrepancy in the number of chromosomes between humans and chimpanzees, evolutionists have proposed that at some point since humans split off from chimpanzees about 6 million years ago, 2 chromosomes fused together into what we now know as human chromosome number 2. This idea first got going in 1982, when high-resolution images of chromosomes from 4 different primates were compared against each other to infer the evolutionary ancestry of these 4 species. The 4 species in question were humans, chimpanzees, gorillas, and orangutans. Now, in particular, for human chromosome number 2 was compared against 2 chromosomes from each of these 3 other primates. Now, chimpanzees were the closest with what is now called chromosome 2A and 2B. Here you can see that the bands in human chromosome 2 match up pretty nicely with what is now called chimpanzee chromosome 2A and 2B. With the banding pattern similar between these 3 chromosomes, it's easy to see how these 2 chimpanzee chromosomes could fuse together to become human chromosome 2. This was further expanded upon in later publications through the use of sequencing. As DNA sequencing became more readily available, later publications would go on to try and find the exact site at which these 2 chromosomes fused together. To find a candidate for the fusion site within human chromosome 2, these scientists look for the repeating sequences that are typically found on the end of chromosomes, the telomere repeating sequences, TTAGGG, or the reverse complements of that. Here we have the 798 bases that make up the alleged fusion site within human chromosome 2. We're looking for the repeating sequences of TTAGGG. On the other side of the fusion site, we have the reverse complement, which is CCCTAA. As we follow the direction of these, we can find out the fusion site, which we have here highlighted as AA. In addition to the fusion site, these scientists claim to have found a second centromere, as the two chromosomes, chromosome 2A and chromosome 2B, fusing together end to end, would have two centromeres. Now, centromeres, like telomeres, have repeating sequences. However, with centromeres, the repeating sequences are 170 base pairs long, not 6. So at this point, the case for two chromosomes fusing together to make human chromosome number 2 seems quite likely. If the scientists here are correct in that these two sites, the fusion site and the second centromere, are as they claim them to be, then it gives weight to the idea that human chromosome 2 came from two separate chromosomes, and by extension giving weight to the idea that we share common ancestry with chimpanzees. However, when one looks at the sequences of these proposed sites, it doesn't really look like what the authors are claiming them to be. It seems very unlikely that they were ever from two chromosomes. Here's why. For starters, the fusion site is way too small. Telomeres, the ends of the chromosomes, vary in length from about 5,000 to 15,000 base pairs in length. With two telomeres together, you'd expect anywhere from 10,000 to 30,000 base pairs in length. However, the proposed fusion site is just under 800 base pairs, about 12.5 times smaller than the smallest that we would expect. Secondly, the proposed fusion site doesn't look like what it should. If there was a fusion between two telomeres, then we'd expect the sequence to be almost identical to an actual telomere. However, given the rate of mutation from parent to child, 60 base pairs per generation, we would expect one sequence change here within 2.5 million years. With 6 million years between a shared human-chimpanzee ancestor and today, we would expect to see 98 to 99% similarity to that of a natural telomere. However, in reality, this site only matches the telomere sequence by about 70%. This leaves us with three options. 1. The mutation rate is insanely high here. 2. The fusion occurred well before humans and chimpanzees split, but that negates the whole point of the fusion site thing in the first place. And thirdly, there was never a fusion here at all. We'll get back to this. In addition to this, the longest repeats of perfect telomere sequences, TTAGGG, is 2. Yet outside of this, in the rest of the genome and outside of telomeres, these repeats can occur 3 to 10 times next to each other. So are they more likely candidates of a fusion site? Not really. No. In addition to this, the proposed fusion site was found to be within a functioning gene. This wasn't just some part of a gene that didn't matter. It's actually important for the gene being used, the expression of the gene as we call it. Gene can be turned on and off in different cells depending on the needs of that cell, and the gene itself in human chromosome 2 is expressed in 255 different cell types, something you wouldn't expect from a happy accident. So since it's within the gene, was the gene present in both chromosome 2A and 2B before the fusion? Or was this a new protein that formed after the fusion? Then the fusion site isn't a fusion site. Was there never really a fusion, and this is just grasping at straws to try and find a fusion site? We actually have a way to look for this. Using a method called fluorescent in situ hybridization, or FISH, we can take the gene of interest, in this case the gene that has the fusion site within it in humans, attach a fluorescent dye to it, and then get that to bind to parts of the chimp genome that have the same gene. If it's not present, then we won't see it. If it is present, we'll see an illuminated spot where the gene is present. Using this gene that the proposed fusion site's in from humans and putting that into chimpanzees, we find that chimps have 2 copies of this gene, but it's just not found on chromosome 2A or 2B. It's found on chromosome 12 and 20. Humans have 18, and they're found in different spots to that of chimpanzees. This means that if there was a fusion between chromosome 2A and 2B to make chromosome 2 in humans, then the gene wasn't present when the fusion occurred. And then the gene was copied over in parts, with the first part before the proposed fusion site and the second part after the proposed fusion site. And the fusion site is responsible for expression of the gene. The gene is actually functional and is functional in over 200 different cell types. Something that you wouldn't expect from a happy accident. Moving our attention now to the proposed second centromere, the part of the chromosome that's responsible for a successful cellular division, we see that this too doesn't match what it should look like. While this proposed second centromere does have repeats, it doesn't match those that are found in functioning centromeres. And to make matters worse, it's human specific. That is, it's not found in chimpanzees or any other animal. And in addition to that, it's not as long as an actual centromere is. It's way too short. Even worse is that it's found within yet another functioning gene. And there really shouldn't be genes within centromeres, or for that matter, centromeres within genes. This is not a centromere. And the other site's not a fusion site, and while evolutionists believe that human chromosome 2 is good evidence for a shared evolutionary ancestry between humans and chimpanzees, as we saw in this video, the proposed fusion site and second centromere aren't what we'd expect to find if there was a fusion between two chromosomes. And with the other details that we talked about, like being within a functioning gene and being way too small for what they're supposed to be, and for the sequence differing way too much for the proposed evolutionary time that's passed, this is not evidence for a shared evolutionary ancestry between humans and chimpanzees. Instead, this is more stronger support for intelligent design. These things were made to perform a function, and they perform that function. If you found this video informative, please give it a thumbs up, and if you know someone that would benefit from it, please share it with them. Until next time, God bless.